Join us.
It's good to see you too.
Yeah, and
Yep,
so we've got a good group here, and I'm just going to introduce you real quickly.
sure.
Carrick is the fruit pathologist with Cornell Cooperative Extension. He does a ton of work with tree fruit and also supports the berry end of things, and I'm really happy to have him. He has a very active um, research program, extension program, and he has a fairly full teaching uh, menu as well. So for some reason, your video has frozen, so hopefully that's not going to be a problem. I'm going to turn my video off, and I'm going to let you go ahead and share your screen
Yeah,
here.
the machine here, Laura, had some trouble yesterday with the Zoom, and I took the camera home, plugged it into a different computer, and it was fine. However, it's been acting up here, unfortunately. I
Why
don't
don't you
think it's a
just turn it
bandwidth.
off? Why don't
Yeah,
you
I'll go
just
ahead
turn
and
the
turn it
video
off.
off? Yeah.
And maybe we'll try a different camera in the future, but I'm starting to think it might be the computer. So I wonder if I turn it back on if it unfreezes.
We had a bunch of
Nope,
little
it is
goofy
completely dead.
things, yeah, that
Very
happened
interesting.
last week. I had a bunch of goofy Zoom things that happened, little
Yeah, and
weirdo
I think
things. So
unfortunately this is the machine in my office, which is brand new for the pandemic. But I guess I burned it out of the ports out or something. It just hates this camera now. What I'm going to do is I'm going to go ahead and share the screen, and I'm going to share screen number 2. And one of the things that's it's also it's nice to be talked at for the entire time, but sometimes it is also fun to get to do some interaction. So what I've done is I have a couple of these different little polls, and how to access them is as follows. You either text this phone number, the phone number 37607, this phrase, and I'm going to paste this in the chat, or you can go on your computer and click on this link. You don't even need a name or a login. And when you get in there, just push an A. And it's just sort of testing us. So as we go along through the NEWA exercise, I'll ask you some questions and stuff like that. And it'll be a little more normal. Now you should see all of this information popping up in the chat. And we can go ahead and fiddle with it. Since it's 8:11, I'm going to go ahead and start. But if you sort of pick at these things when we get to one of the polls, I'll show you, and you can kind of look and see what other people are thinking, see what you're thinking, and when you get to them, and it will look like my picture of my phone if you log in, or it'll look kind of like this other picture of your screen in the background. And all you do is you just hit an A, B, or C in many of these cases, or you type something in. But in our case, this is super easy. You just, in this case, for this example one, you just type an A, and if you're on your computer, you can see what the silly question is. But we'll have some relevant ones very briefly. So that all the instructions are in the chat. You can keep following along, but I'm going to go ahead and get started, and we'll let this go. So the ideas for the day are, and I'm going to go into full screen mode, and let me know if also my microphone pops out, because one of the things that also happened the other day is that my mic was cutting out, because I think it's the USB ports on the back of the machine. So if that happens and you can't hear anything, type something in the chat, and I'll take this machine and I'll try to flush it down the toilet after we're finished with this particular webinar.
everything so far
So
sounds
far,
good
so
and
good.
looks
All
good.
right,
Yes. Yep.
great. Well, so first, the first thing we're going to do is we're going to try to understand the concept of disease forecasting, how it's used. I'll show you the NEWA strawberry model, and I want us to get into an exercise with the NEWA forecasting system so that you get to try it for yourself. And I'm going to give you all the ins and outs on how to do it. I may skip a couple of things just to save us time, because I don't want to cut into Marvin's time too much. And we're sort of moving. So the idea is that all of these scientists have done all this epidemiology for so many years. And what they do is they often yes, and with my weather data, I can predict the risk and occurrence of disease before it has occurred. This is great because you want to spray fungicides before disease develops. However, what they've never done is they've never really tried to put it into a system where it might show up on a grower smartphone or a web browser. And all of this information has been known and done for many, many, many years. It's just that no one's bothered to make it accessible, because epidemiologists like to wear little bow ties and sit in their offices and drink lattes, and we talk about things. But now let's try to bring it to the real world. And so it uses phenology, pathogen presence, and environmental conditions all working together to sort of live guess when the problems are going to be. And all of this has been historically accepted and validated scientifically, but now what we want to do is make it into simple web-based forecasting tools. And they're used in agriculture worldwide. Everybody else is already doing it, particularly in the row crops. They've been doing it for years. And now, finally, some trickle-down effect. We've been able to cob enough little money here and there to sort of do this ourselves, thanks to the the New York Berry Growers Association, who's pretty much funded everything we've done with this model and actually allowed us to actually bring it to you. So why do you want to do it? It really helps increase the precision of your fungicide application. The fungicides work best when the pathogen is present and when the disease risk is highest. It's like, when you wear your seatbelt on the thruway and there's no traffic and nothing, the seatbelt isn't doing much. However, if you're going into like Syracuse at 5 o'clock in the afternoon, coming back from one of these meetings when we could drive to them, then that's when the seatbelt has the best chance of working, because something crazy will happen, at least at some point in the drive. So this really increases your cost effectiveness of pesticides. It also will allow you to not avoid putting out a pesticide when there's no problem. It promotes pesticide stewardship and saves you money. And then finally, if you really put them on right when the disease is there, you end up, you prevent a lot of loss due to disease. And NEWA has a whole bunch of models. So even if you're here doing grapes, onions, potatoes, tomatoes, and turf, they're all there. And they're actually fairly good. And oh goodness, if you've actually gotten into the poll and you're curious, I'm going to activate this one and we can see if you use it. Would you ever consider using it just based on the silly information I've told you now? Let's see if anyone, yep, a lot of people guessed that they were nature's greatest miracle. What I'm going to do is going to pop back to these activities, and we're going to turn on the one about would you use disease forecasting. I'm going to activate this, and then you can begin to like push some buttons, A, B, or C. Yeah, I'm sticking to my calendar schedule. Maybe if it's not too time consuming. And I do not get offended on what you put the answers in. I'm too tired to worry about those types of things. So if you don't want to use it now, and number C is the one I often think about when I go through stuff. Maybe if it's not too time consuming. And in that case, let's see how we can make it less time consuming. And maybe if it's a little bit easier. All right, so let's look at what the NEWA model is and talk about it in terms of disease forecasting. What it works for is fruit rots, and you have things like anthracnose, and I'm not going to spend a lot of time on them because I'll talk about them in the other talk a little bit. But they rot your fruits, they blight your crowns, they cause leaf spots, they do wilts, they do deaths, and they cause these sunken lesions. They can kind of look blackened as over time because they get kind of stromatic and dark. And if you stick your finger in them, there's a bunch of gross spores, and it can be kind of orange if you catch them in the right frame of mind. But where these models use is these things like warm temperatures and rain. And if you're in the Albany area, or even a high relative humidity, you might have a greater chance of getting these than compared to like maybe the Lake Champlain area. And they do like artificial irrigation. You think, well, I'll cover it and get rid of it. But what ends up happening is like drip near the soil line, high relative humidity under the canopy can end up causing infections and dispersals as well. And what these models do is they tell you when these times are. And so you put the fungicides out when the right amount of warm rain has happened. Oh goodness, this looks gray mold. We've all seen this ugly thing before. Usually, hopefully shows up after you've picked the fruit. Soft rots. You can even cause a shoot blight. It can be really bad, such as in this this tabletop strawberry right there. And as you can see, it got on the soil line and just went to town and rotted the fruit. We want to avoid this. What do you know? The epidemiology is pretty much very similar. Warm temperatures, rain, or high relative humidity. Can we use this to make a model? And many, many years ago in the 90s, when I was a small child, they did. And no one has ever brought it. But recently, thanks to the New York Berry Growers Association and all of their efforts to get a lot of funding for research dollars, Julie Carroll and I were able to work with the people in Florida and Maryland and make this a reality. And so we have one for fruit rots. What does it do? Well, and we'll go through it, I hope. But it really predicts favorable conditions. It just tells you when it's risky. Think of it as your the traffic alert on your GPS or your phone that's like, oh, I'm going into Syracuse, it's 5 o'clock, and it's, warning you of aggressive drivers, those types of things. What this does is it warns you of possible infection events. And if your seatbelt wasn't on, now's the time to do it. Definitely. And what does it do? If you have a weather station at your farm, it will use your local data. If a rampaging deer goes tearing through your operation and trashes that weather station, it will still pick up the satellite data from the most closest location. There is gridded data that is also filtered into it. And what it will do is it will pick up leaf wetness algorithms and other stuff and will begin to predict various risks. And so this is kind of what it looks like. It's very simple. It doesn't give you a lot of explanation at all, but that's what I'm here for today. There's only one disease to pick, I think, and I think it's strawberry diseases. You get a state, it has all of them in it. It's got locations. They're called station, but it's really sort of location, and you can find the one closest to you. You can pick the date of interest, and it will give you some risk levels, no, moderate, and high. And to be very I've been using it for a couple of years in my research plantings. I usually spray when it's high, unless I haven't sprayed in a while. And we'll talk about how to use it to actually plan your applications. And you can kind of see here, it gives you the day of interest, and then we'll tell you many days into the future. Now, the one thing that's nice about this is it uses the forecast data very conservatively. That way, rain often is predicted pretty heavily 3 to 5 days out. And what the NEWA system says, I'm not going to put as much emphasis on that rain. I'm going to really worry about the rain 1 to 2 days out from the prediction. And then in that particular instance, it sort of weights everything a little bit differently. Because as we know, you can never really predict the water or rainfall up until almost the day before it is actually happening. Sometimes with lake effect snow and stuff like that, you can't really predict it at all. So that's how this works. And so we're going to run through an exercise, and I want to make sure we get to at least get one of them in before we get going. It's going to predict a lot of infections, and this is where the critical part comes in. But you want to apply at the highest risk period, not every infection. You're like, well, how do I use some real rules to determine when I should spray by this? And this big blobby blue text is the thing we want to pay the most attention to. If you have not sprayed in 7 days or had an accumulative 1.5 inches, what I do, and I picked on this from Kathy Dempchek, and she said she used it, and it's made a lot of sense, and it's worked really well in my research trials. All right, let's say you put on a spray, and then you wait about 7 days, or if you had more than 1.5 inches of rain, you start looking at the model and looking at the forecast of the model. And when you see it turn red or give you that high risk period, that's when you want to get your next spray on. You don't need to spray every 7 days, but what you need to do is you might start looking in the future. Or if it's just you sprayed and it just rained 1.5 to 2 inches, you might start looking in the future. And that's when you might want to think about applying before that next high risk period. And anytime use common sense. If it doesn't look like it's ever going to rain, or the model data doesn't ever look like it's matching up with your location, for goodness sakes, don't use the model. It can be just like the Garmin, but for the most part, it's working fine. But if something doesn't make sense, always use your common sense first. What the other thing to do with this is don't spray after an infection period. This is one of the few practices that selects for resistance. And that's something we don't necessarily want to do. And, you might ask, what about other diseases? I can't just spray for anthracnose. What about my powdery mildew? What about my leaf spot? I've shown some leaf spots here. We don't need to worry about what they specifically are. But what's interesting, as I've noted about this, is they're all in the same group of fungi. They're all the same type. You know what they all like? They all like warm, wet water. And I've also noticed there's often more variability than the environment, than the genetics of these various organisms. So even if you're using the model to spray for your fruit rots, I found that it works actually pretty well for some of these other diseases as well. They're all very highly related, and you get them. One's maybe a wolf, one's maybe a dog, and one's maybe a chihuahua, effectively. But, it gets them all because they're all carnivores for the most part. So that's how that works. So let's jump in and do an exercise. And so we're going to do one together, and then we will end up trying it. So the first thing you want to do, and I'm going to try to see what I've picked here. I'm going to paste this link in the chat box so that you can actually go to it. This is the model. It's not very NEWA friendly right now. They have not changed the model. They put it on the NEWA web page, but it is literally still at GitHub. So in that case, I'm going to click on it. Click, and then I'm going to move over to my web browser, and you do it as well. And hopefully you'll see this following screen. I'm going to go away from this, and we're going to check out the strawberry model. Okay, so what do I want to do? Hopefully this will come up. I will wait very briefly, but I don't want to take all of Marvin's time. So let's pick together a scenario. Now, the model behaves really weird when you're asking for forecast data and it's back in time. If we use the model now, February 2021, it's just going to tell us a bunch of nonsense and you don't need to pay attention to it, because unless you're in Florida, there shouldn't be any strawberries in the ground. So I'm going to pick state New York. Fine. I'm going to pick Albany right there. And if you just go to the pull down menu, it will pick them all. I'm going to also try to magnify my screen so that it looks more normal. When you have these really big high resolution, well, maybe 300 is a little much. There we go. Let's see if that's a little bit better. So let's going to pick Albany. And if you've already seen the thing, we want to pick August 1st, 4th, 2020. And just know that when I pick August 4th, it's not going to start with August 4th because it's behaving strangely from the forecast data. So I'm just going to pick that. Let's do, in my case, I always do a lot of day neutral, so they're always in play at this particular time. And hopefully this will come up on your screen. And you can begin to see that it's July 28th, July 29th, July 30th, and you'll see a lot of green. You see some yellow warning periods, and then you see one coming up on the 4th. So I'm going to go ahead and move to our next scenario in this particular instance. Now, if anyone's stuck, paste it in the chat. We can see it. Very good. I'm going to go ahead, and now I'm going to pick this up. So this is where we are. Here's your scenario. You last sprayed on July 28th. Okay, it's clean. It's July 31st now. There's been no rain. And then the question to you guys for you to consider, there's never a wrong or right answer. It is, when do you spray next? When do you wait to spray? You could say, do I wait to spray? Do I go August 1st? Do I August 4th? Do I go August 2nd? You can look at the model and see when you think you might actually do it. There's a big day coming up on the 4th. And what I'm going to do is come back over here and we're going to let you all chat about what you want. When you would spray, I'm going to go to the bottom of this big list here. Oh no, is it gone? Did I move it? Well, it looks like for some reason my poll isn't showing up. But if you want, you can just post in the chat when you think you would spray in this situation. Would you do it? You sprayed on the 28th. August 4th says there's another big one, but there's been no rain. What do you do? What different things do you do? In my case, if I was doing a research trial, and it depends what you have, maybe 8.3 gives you a lot of play right there. And it also, another thing that can happen with fungicides is light and water can degrade them, and then they can break down. In that particular instance, August 2nd or 3rd, you could wait to spray, but it's a pretty high infection period. And what I would do, and you'll notice this model doesn't have the weather data, I would go out and I would check the Weather Channel and a couple others and make sure that everybody was saying, oh, big rain is coming. Let's take it through another scenario. Now, if you can, right before we quit, go back and then go back to your model and then click August 10th. And this is a weird scenario. If you run it in real time when the season is happening, you can begin to see stuff. So theirs is a different one. You know now that it's August 4th, and let's just say you sprayed. And we'll say in this case, you got the spray in before this big rain, 0.7 inch of rain on the 2nd. And, hey, great. And you hit this, and now it's August 4th. This rained here, and maybe there's a little bit more. And then you're looking again at the thing. And do you think, do I spray again? There's some light weather coming up here. This is where it gets tricky. And this is where your extension person can use these models as well and say, well, if you sprayed on the 2nd, do I need to worry about the 7th? Do I need to worry about the 10th? How much rain is that? What do you do? I mean, there's no wrong or right answer. You can post in there what you think you would spray on. And so, and in this case, this is where it gets a little bit tricky. And it should show up on your phone, and you see it resizes. And if you make it too flat, you lose the sidebar. But in the end, again, this is where you can begin to think, do I spray again? I already did it on the 2nd, but I got a 7/5 of an inch of rain. What you might do is you might think about what fungicide you have or what fungicide you might be doing. But the model's fairly simple. It's fairly responsive, and hopefully it doesn't seem to crash as badly as some of the others. And in this particular instance, you had a big one right there. You might have gotten some more wash off, and you can decide again whether or not you would spray again. Some people might spray on the 8th to watch out for the 7th. And a lot of it depends on what you use. If you're going full organic and you may be using a biopesticide or something like that, you might think, well, I'm fine. What you might do is you might also look at the Weather Channel, and one day if we had the programmers here, we could actually put the amount of rainfall predicted on all these days. And then you could look ahead and say, well, is it going to be 1.5 inches of rain? I'm not sure. Oh, if it is, then maybe I will spray on August 8th or 9th. And so some of those things are the things to consider. And it's a fairly easy thing to do. What I'm going to do now is we'll take any questions and I'll jump ahead and give you some acknowledgments. If you want to ask any questions, you can paste them in the chat or you can say them out loud, I suppose. Very
Thanks,
cool.
Carrick. I just want to invite everybody, if you want to put your questions in the chat box, we could actually unmute if we don't have a ton of conversation all at once, but maybe letting us know if you have a question, and then we can call on you. I just have one comment about so the model is only as good as the data, and in some respects we have some challenges, especially in Eastern New York with our weather stations and locations of weather stations. And so I just want to caution people that you really want to figure out where your closest weather station is. And if you are really intrigued with this, you can get your own weather station. We've had lots of information sent out in the past about how to get a weather station and how to, kind of get into this NEWA system with your own weather information.
Yeah, that's a good point. And that's why I also recommend, if you're looking at a place that's not close to you, also check your own local weather data or whatever weather service has been providing you what you believe to be the best data. And, seeing if those red things match up with a lot of rain and warm weather, because that's in theory what they're doing behind the scenes. Yep. Good.
Anybody else have any questions?
Right. I'm going to go ahead and stop the share and then not turn my camera on. Well, it's all dark.
And I think that one piece that you said, Carrick, about not following an infection period with, trying to avoid resistance is huge. And I don't think we talk enough about that. And spray timings, we're usually only talking about chemical class rotations. And, that's a point really well taken. So,
and I guess in that case, I should also mention, if you're using a biopesticide or you're using Captan, I mean, you don't get any post-infection activity, but those things won't get resistant. So you're like, I'm going to use Stylodyl. I don't have to worry about resistance. No, in that case, you don't have to worry about it. I want to use, Serenade Opti or Double Nickel. No, you're fine. But you just won't get as much bang for your buck, because what Captan does and those other things are just better protectants, and they like to redistribute a little bit. But if you're using Switch or Roverol or Maravon or Pristine, those are the ones that you definitely want to wait and put them on first and not afterwards. Even though they do give you some post-infection activity, that's where you might lose it.
All right, great. Does anybody else have any questions at all? Okay, so there's a couple of things that are coming in privately to me. How accurate is the model compared to a standard spray schedule?
That's a good question. And actually, if you want, I'm going to show you what it looks like compared to a weekly one in disease control in the next talk. I prefer to use it. There's no official DEC polls. They're just for fun, just so that you're not completely bored out of your mind. I'll show you in the talk how it works. I often get away with the same level of disease control as a calendar or better, and having a lot fewer sprays. And so you can check the next talk for yourself and see what you think of that.
Any other questions? All right. Seeing none, Carrick is going to be around, so if something pops into your head, we can still get that at the end of the session. So Carrick, thank you so much.
You're welcome.
And our next speaker is also going to be talking about some diagnostic tools that are available for us. And Dr. Marvin Pritz, I think most of you who have been involved in fruit crops for a while know Marvin, but he is the horticulture specialist and faculty member at Cornell University. He leads a very dynamic research program and is also very active in guiding undergraduates and graduate students. And so I want to welcome Marvin.
Thanks, Laura. And good morning, everyone. What I want to do today is share with you a way of thinking about diagnosing problems that you might see in the field. We often get calls, I get calls, people send me photos of things, say, what's wrong with my strawberry plants? And I always follow up with a bunch of questions. And what I want to help you with is like help think through how we would go about figuring out what's wrong with your plants, because it could be a disease, you know, some of the things Carrick talked about, but there's other things that could affect it as well. So I want to go through sort of like the process of narrowing it down and figuring out what's wrong, and then what tools can we have to help you solve that. So I'm going to start here. So diagnosing strawberry problems. So a lot of things cause problems, and I've got a whole list of like a dozen things on the left-hand side there that can cause problems and cause symptoms in your plants. And the problem is a lot of these things look alike. And the photos on the right-hand side here are two images of something wrong with the strawberry plant. Symptoms look kind of similar, right? The younger leaves are kind of starting to turn yellow in both cases. We're starting to get some death, necrosis on the outside of the older leaves. The same thing will happen here in a few days. So what is it? Is it a disease? Is it some sort of toxicity? Exactly what's going on? So this tool is designed to help you figure that out, right? And I'll tell you, like in this case here, the top photo is Roundup drift on strawberry, causes these yellow leaves. And over here, this is too high of a pH, and it causes the younger leaves to turn yellow because it's iron deficient. So two very different things, herbicide injury and high pH causing very similar problems. So just looking at the picture doesn't necessarily tell you what the problem is, or give you clues. I mean, it's clue, but you need more than that. You got to look for like larger patterns. And so that's what we're going to be talking about. A couple of more examples over here on the left. You see these strawberry leaves turning brown and kind of becoming necrotic. Same way down here. Kind of looks similar, but again, two different causes, very different causes. Up here, we have some herbicide injury. And down here, we have leaf scorch. So we have an herbicide and a disease. The same way over here, we have leaves curling up, and we have leaves curling up. In this case, we have powdery mildew on the underside of the leaves causing leaf curl up. In this case here, we have herbicide causing the leaves to curl up. In this case, stinger. So there's several steps to figuring out how can you distinguish between these causes. And here are the steps you sort of have to go through. The first thing is determine if these patterns that you're seeing are normal or not. Sometimes people will send a photo of something and it's like, it's a normal thing. It's nothing to be alarmed about. It's not a problem. So over here on the upper right hand, you see these strawberry leaves with, you know, water on the edges, these drops of water. And if you're not experienced with strawberries, you might think, oh, there's a problem here. What's going on? That's just natural guttation, exudation of water out the pores of the leaves at night. And it means that your plants are well watered and healthy, nothing to be concerned about. If it's abnormal, if it's something that you normally don't see or expect, you really need to make note of when did the symptoms first appear, because that is a very big clue as to what's going on. When you call in or talk to somebody about your problem, not only note when the symptoms first appeared, but a little bit of history, like when did you fertilize last? What did you spray the last few times? Was there any extreme weather recently? And make note of that, because those are all hints and clues as to help us figure out what's going on. And again, if you look over here on the right, we're all familiar with these black centers in the strawberry flower that indicate frost damage, but again, someone who's inexperienced might not know that and might say, hey, I've got these black centers in my flowers, what's going on? Well, what was the weather like? And you can figure out when the frost occurred. But probably more importantly, we want to look for patterns, both large-scale patterns in the field and patterns that are on the plant itself. And then we want to match the symptoms with the possible causes. And we'll have this diagnostic tool we'll show you to help you with that. And then lastly, in some cases, you can get a diagnostic test. You can send the plant in for analysis, leaves or soil, or look for nematodes in the roots or viruses. It's sort of a way of confirming what you might suspect. So let's look at the field scale patterns. First of all, let's talk about that a little bit. You have a problem, you look out in your field and see, is this problem following the variety? Does it change when one variety changes to another? Does it follow the soil type in the field? If the soil changes, is it at the bottom of a swell, every fourth or fifth row, just along the edges? Is it random? I mean, those are all clues as to what's going on. And if you look over here at the photo, well, it's pretty clear that this is a variety thing. So on the right, this variety is resistant. On the left, it's susceptible. And knowing what the varieties are here, it's able to pretty clearly tell almost right away that this was red steel. That the variety on the right was resistant, the one on the left was susceptible. But notice it follows the variety in the field, and that's a really important pattern to watch out for. Soil type, if you see a pattern in the field that follows the soil type, that usually indicates that it's either a nutritional issue or a disease issue. Because a lot of diseases, particularly soil-borne diseases, like sort of wet, damp conditions. And if your soil type changes from, say, a loam to more of a clay, you start to see the patterns in the clay part of your field, that might be a pretty good indication. It might be a root disease. Same way if it's the bottom of a swell where water accumulates, makes it a little bit wetter, that's often an indicator that there might be a disease problem. Or, you know, cold air sinks down low too, and if it's something, if your plants are in the springtime, you get a lot of damage low down in the swell. It could be from cold temperatures where the cold air settles in that swell. If you see something that looks like a regular pattern, like, oh, this is weird, like every fourth row, the plants are turning yellow, what's going on? That's a pretty good indication that it might be a spray problem. You had sprayer overlap. So when you turned the spray around and came back, there might have been overlap on that every fourth row causing the symptoms. Or if you see the damage at the ends of the rows, oftentimes if you slow down your tractor to make the turn, the sprayer is still pumping out at a constant rate, and you can overdose the plants at the edges of the rows. Oftentimes too, insects come in from the edge of the field into your field, and you can see a gradation of symptoms from the edge to the center. And then if you see random patches, that's often due to the viruses that are spread throughout the field and then start showing up their symptoms sort of in patches and not like real even across the field. So those field scale patterns help you figure out what's going on with the problems that you're seeing. The other place to look for a pattern is on the plant itself. So we often ask, well, do you see damage like just to the older leaves? And if the answer is yes, then that's almost always an indication that there's a nutrient deficiency. Because in plants, there are certain nutrients that are very mobile in a plant and those that are very immobile. And if you have a deficiency where the nutrient is not very mobile, you're going to start to see symptoms show up in the youngest leaves first, because the nutrient doesn't move into those younger leaves. So they're showing the deficiency. Conversely, if you see it in the older leaves first, that usually means the nutrient's very mobile and it's moving to the growing points but causing the older leaves to become deficient. So that's a big clue as to what the nutrient might be that's deficient. If you see it on older leaves, usually the deficiency is NP or K. If you see it on younger leaves or the extremities of the growing points, it's usually calcium or sometimes iron. Herbicides also move through the vascular tissue to the extremities at times. So that could be that too. That's where it's important to go back and look at your history. What was sprayed? Could that be causing the symptoms you're seeing as well? Uniform damage, often from spraying. And then are the symptoms in the roots, leaves, flowers, or fruit. And sometimes this can throw you off, particularly if you're looking at above ground symptoms only and not looking at the roots. Because if the leaves are wilting, that often doesn't mean there's a problem in the leaves. It could mean there's a problem in the roots. More times than often, that's the case. So you want to dig up the whole plant and look and see what's going on underground. So just because the leaves are wilting doesn't mean there might not be the cause happening under the ground. So look for the problems in the field as a whole, look for patterns, see if you can relate those patterns to the variety of the soil, and then look within the plant itself and see what could be causing the problems. So once you have these sort of patterns figured out, then we have this tool that we have for you to use to help to try to narrow down what those symptoms might be. So I'm going to jump to another screen here. And this is our diagnostic tool. And the URL was on that last slide, or you could just type in Cornell Berry Diagnostic Tool. And this is what you come up with, this page. And you can click on the crop you're interested in, and we'll do strawberries. And then it asks you a bunch of questions about where do you see the symptoms. And then by clicking and answering the questions, it narrows down what the possibilities are that might be causing the problem. So for example, we'll start with this first one, the whole plant. Plants are wilting and collapsing, and it's a young planting. So what could be causing plants to wilt and collapse in a young planting? Well, we go in there and we see, well, the plants are wilting and collapsing. Is the outer leaves first? Yes. So we can click on that, and then we can see what the possibilities are. Verticillium wilt, very characteristic. In young plantings, the outer leaves die first and leave the younger leaves. We say, no, the individual plants were suddenly collapsing and dying, the whole plant, not just the outer leaves. And there was plenty of moisture. What could be going on there? Well, Anthracnose could be one, Fusarium, Pythium, Rhizoctonia could be another. You say, oh no, the crowns, the growth is uneven, the crowns are brown in the interior. It looks like if this is a new planting, the plants could have been stored at too cold of a temperature when they were planted. So you have this damage to the crown. And the roots are stubby and chewed. When you dig them up, you see these grubs. Well, that's probably what's going on and causing the planting to, and a young planting to wilt and collapse. So this gives you lots of possibilities there and gives you photos to help out with that. Let's look at an older planting. We can see some choices here. This plants, most roots are black. The plants collapse near harvest. We can dig up the roots and look. We can see the roots look like this. We scrape them away. We see this red core in here. That's pretty typical of Phytophthora. And we can see these fields where the damage is infected with red steel. So, and then also at the bottom, it gives you some other resources you can go to for more information. So let's just go do a couple more examples. Let's go to leaves and stems. The leaves look funny. They're colored or spotted necrotic. You can click on that. Leaves appear yellow. Let's see what could be causing that. Oh, there's a bunch of choices here. Does the yellowing appear suddenly? Yeah, let's see what that could be. That's the one we saw before, Roundup injury. Sprayed Roundup, the drift, all of a sudden the leaves started turning yellow. That's a possibility. What about yellowing appears suddenly, it's distinctly veinal. Too much Synbar can cause that. Yellowish tinge at the margins of the exposed leaves. See this? Caused by leaf hoppers. So the beauty of this tool is it puts everything together in one place. It's not just diseases, or it's not just insects, or it's not just herbicide injury. It's all these in one place. We can help narrow it down. Just do a couple of more. We'll go back to, let's go to flowers. Let's see, flowers are cut just prior to flowering. And I don't know why that's not doing that. Should be a bud weevil. Oh, there it is. Strawberry bud weevil. And we can go to fruit. And let's say our fruit is deformed. What are some possibilities there? Tarnished plant bug can cause that. Frost injury can cause that. Boron deficiency can cause that. Poor pollination. And then we got this weird thing here called phyllodes that nobody really knows what causes that. Oftentimes, some people think it's a mycoplasma. It's like a virus-like thing that causes that occasionally in the fruit. So this tool is for you to use and to help figure out what's wrong, or help you figure out what's wrong with your plants. And we have this for strawberries that we just went through, but we also have it for blueberries and raspberries as well. And we just updated it, so it's pretty current. So with that, we'll see if there are any questions.
There is a question for you in the chat box, Marvin.
Okay.
About the length of a productive, maximum number of productive years a planting can be kept.
Yeah, so typically in New York State, I think growers fruit a field for 2 to maybe 3 years is typical. We had a strawberry farm one year, and there was this one field that just kept pumping out fruit, and we just didn't. I think we fruited it for 5 or 6 years in a row. That's pretty unusual because most fields didn't last that long. But every once in a while, you get a winter, and it just keeps on going. But I would say typically it's 2 or 3 years, and usually the reason you take it out is because the crowns on the strawberry plant keep dividing, but the root system is basically the same root system. So you get more and more flowers per root system, and the size starts to decrease. So the berries just get small in older plantings. And then also it's harder to keep the weeds out. So the fields just get weedy enough that people just give up on them. And some organic growers I know, because they don't have the herbicides, they just assume that they're going to fruit a field just once or twice and take it out. That's just part of their plan, the rotation. Just do it a couple of times. But I think if you're willing to use herbicides, you could probably get 3 good years out of a typical strawberry field.
Great, thanks. Does anybody else have any questions? I do want to just say, I mean, what Marvin just talked about is what we are like every day of my life. This is what we go through. So this is a really important topic, is just trying to ferret out what is happening in a field. And it's really hard, but the more information you guys can provide to me, to Marvin, to Carrick, the better. So regular observation is really important. Like maybe you're not going to see your field every day, but you know, you might definitely want to try to look at it every couple, three, at least weekly, kind of walk through and not just look at it when you drive by, but because there's stuff happening there all the time. And the sooner we see it and the sooner we recognize the you know, the pattern, that is very helpful. Once everything is really badly affected, sometimes it's easier. We know right away what happened, but there's very little to do for it. So that's just really important. So I really appreciate this information. And the other thing I will try to share with folks that get the newsletter, our colleague Elizabeth Buck did a really good little kind of a summary of how to take a really good diagnostic picture. That's super helpful. Some growers really get that beautifully, and they're kind of avid photographers, it seems like the pictures they send are fabulous. But it's really helpful to have a picture. It's very difficult sometimes to talk about the symptoms and understand exactly what the grower is saying. So smartphones are wonderful. And if you can send pictures, that's great. So Marvin, I thank you very much. Before we go with Carrick's topic, we need to release the DEC poll, and there's only 4 really quick questions, but if you want DEC credits, you have to answer this poll, and we have to have a recording of you answering the poll. And those of you who don't want DEC credits, we're kind of interested in the results. So go ahead and do the poll. We won't be keeping this for any kind of record. It's just, you know, we just try to get a understanding handle on where everybody is and learning about some of these tools. Natasha, I'm going to let you just end polling whenever you think you need to, but it looks like people are still doing a little something.
Yeah, I'll probably give it another 30 seconds or so, and then we can.
Yep, we've got time, so that's great. I also want to, at the end of this, I'll put my email and my phone number in. For those of you, I don't know some of the folks, I know a lot of people on the call, but I don't know everyone. And if you're in the 17 counties in Eastern New York, by all means, if you have a berry question, please reach out to me, either email or call me. During the winter, my cell phone does not work well at my home, but I'll try to get back to you as soon as I can. And while we're
go ahead and end this.
Okay,
That's
great.
2 seconds. And okay.
Oh, and Angie, thank you. Angie is giving us a little hint about the submission. When you submit a diagnostic sample to the lab, don't skimp on the case history. So as much information as possible is really, really helpful. Strawberries are notoriously difficult to kind of plate out and actually definitively capture the fungus or the disease problem, especially if it's a root-borne disease. So that's a problem, and the more information, the better.
I think we're good to go.
Great.
Most people have access to NEWA in the past, but not by much.
Wonderful. A little bit of a, yeah, that's great. It's a great tool. And Carrick, do you know when the NEWA, the new website and everything is going to be up and running?
That's a good question. I don't have any idea, but I based on everything I've seen with the pandemic and the speed at which things occur at the university, I would suggest I don't think it's going to be this season. I mean, if it was, that would be great. But I don't know. I haven't heard much. Everything seems more challenging these days.
Yeah. Well, if it does get up and running, it's such a nice, easy platform that I think that would be a good thing, but we'll just
you know, at least it won't change.
Right.
So that's good. So you won't have to learn anything new. I mean, it would be nicer to have the new one, but you might have a learning curve where you make an account and it tracks your stuff.
All right, Natasha, that looks great. Did we have those other 2 questions?
No, it was just the one as far as I can tell. Oh, wait.
Okay.
Aha, found it.
Yeah, I just launched question 2.
Okay.
And then I think it looks like people are pretty, I also had the same similar questions just for the disease diagnostic tool. If you can see them. So it does seem like people see the advantage or the benefit of NEWA. Oh, there's the berry diagnostic one. Good. So we just had 4 little questions, and that's really nice because it looks like most people did not know about this tool. So that's really great that we had Marvin on to talk about it. And the last question is about whether you think it's beneficial, and I hope you think it's beneficial because knowing what you're dealing with is really important to successfully managing the problem. So that's our final question. There, did I bias you all enough on that answer? All right, so I think without further ado, that we are gonna have Carrick start the second talk.
Yep.
And he's going to be talking today about choosing effective fungicides. So this is really important, and I'm sure a lot of people will enjoy this. So thanks, Carrick.
Yeah, I gotta must exit full screen so I can share screen. Here we go. We'll get this thing there. I want to push some stuff out, bring up the chat log, all that other good stuff. I switched to a different camera that doesn't work with this computer, but because you can see it's very washed out, but so far it seems to be holding steady. Even though I went home with the old camera and ran it for 2 hours, not a problem. But that's how things work. All right, so looks like we have plenty of time, and by mandate of the DEC, I have to talk all the way until 9:30. So I think you'll get to hear a lot of things in this one. We'll talk a little bit about biochemical management for berry diseases, and then I'll show you research. And I think we're even going to get to the part where I and show you using the NEWA model to plan all my applications. I noticed a lot of my colleagues do it around the country when they run their efficacy trials, and I started doing it as well, using some of the same guidelines I gave in the last talk, and that's been really working out. Okay, let's see. All right, so there are 2 different ways you can do biochemical management, and I call it biochemical because you might use a biopesticide, you might use a fungicide. And the first is to protect a plant from a pathogen attack using an agent. You think of this as putting on the spray before you have a problem. The second, and depending on what you have or using, they have protectant and eradicant properties. The second will eradicate the pathogens present, just there sporulating on your fruit and leaves. And that's sort of to stop the existing infection and to prevent infection of the neighboring plant. If you want to think of these in terms of common everyday household items, I always like to show this picture here. You can think of your protectant as being the Band-Aid. You got a cut on your hand, you just literally putting something over it to block it. You're just trying to prevent any germination or infection by bacteria or fungi. You can think of the liquid Band-Aid as being a little bit of protectant and eradicant, because it not only does it cover the wound or protect against the part that couldn't get infected, it also provides some sanitation by sort of killing out organisms that are in the wound. And then finally, we have maybe you can think of the antibiotic ointment. It's oily. It will also sort of cover and prevent new things from getting in and infecting your hand or whatever you cut. And also, it has an eradicant in it. And finally, you can think of things just like isopropyl alcohol. That's just purely an eradicant. And when you start to see things, some of the biopesticides like hydrogen peroxide and some of those others, and bicarbonate, you can kind of think of them as your eradicants. This doesn't stay around very long. If you put it on your hand, it's gone a few minutes later. And that's sort of how those types of things work as well. And when you think about the conventional fungicides, there's a couple of different options. The first ones you are most familiar with, and we've had a lot in the past, are multi-site fungicides. You're not going to get resistance from these. These are nearly always protectants, things like Captan, and we'll talk about them. If you ever notice you have one of these, you'll always be applying it at a larger quantity than some of the others. They go in more in pounds to the acres, not ounces. They're broad spectrum, multiple modes of action. Less safe for non-target organisms. If we want to talk about Captan for the example, if you get it inside the plant, it'll kill plant cells. But the idea is you keep it on the outside, you don't solubilize it, you don't put it with a penetrating surfactant, and it kills the stuff out on the edge of the plant. Or Mancozeb. Really low risk of resistance. You'll also notice that many of these multi-site ones have longer REIs, reentry intervals, or longer PHIs, or other things like eye irritation associated with them. And some of the easiest ones you can think about would be Captan, Mancozeb, and even copper is another really good one. There's no real picture of copper, it's just Cu. So we can just say copper in that particular case. And sulfur, these are all these. Now, in contrast, you even have these single-site fungicides. And what they're going to do is just only go at one gene in the fungus of interest. They have protectant and post-infection activity, but as Laura and I were talking earlier, we don't want to always rely on the post-infection activity. This gives the fungus a chance to get established in a big, high number of individuals, and where one of them might have resistance. So while it has that, we don't always want to use it. You'll notice these will go out in ounces, like ounces to the acre, ounces to the 100 gallons. There's really only one mode of action. They're like an antibiotic, and organisms can get resistance to them. They're safer for non-target organisms because, you can get in the plant and there's no binding target for this particular fungicide. Get them on the ground, they don't harm any bacteria. They don't harm any other creatures that are in the environment. And you'll notice that they have really short REI and PHIs, but they will be high risk of resistance. It's just that one target area that a mutation can occur naturally and end up being the point of selection for resistance. And then you might think of one of these fancy ones as being fluxiperoxide, which is an active ingredient in the fungicide Maravon, and has all of these different. Let's stop to move that pole thing out of this. Oh, it won't stop. It keeps springing in front of the thing. Let's see if we can close poles. Yep, as fluxiperoxide in the fungicide Maravon. Now there are other things too that really fall into the biopesticides, and we'll talk a little bit about these briefly. What are they? There's a whole bunch of different types. They're all naturally occurring or naturally derived pesticidal substance. They can be a plant extract. You can think of all those oils of thyme, cinnamon oil. They can be weird minerals. They can even be microorganisms or the product the microorganism makes in fermentation that's antibiotic related. And they can even be transgenes. Even interestingly enough, the EPA refers to the transgenics as plant-incorporated protectants. We don't have a lot of these in berries, so you don't have to worry about that necessarily. But how these things work, they vary in the type. There's all kinds of different types. The costs, usually fairly expensive, and their efficacy, oftentimes one might find to be lower. But they're also multi-site, so they're low risk of resistance selection in almost every case. You can find them all here. If Laura passes out the actual slides later, I have all the links to the lists of biopesticides. And there's another program by the FDA called Generally Regarded as Safe. They're food additives that have pesticidal properties. And so they may not have registrations and labels, but if they're on the list, you can do it. And then there's even another list that has a lot more of your essential oils. They do hurt diseases, but they may not last long. They may not persist. They may not be formulated. And they may not have submitted a lot of efficacy data to the EPA as with a standard registered fungicide or even a registered biopesticide. So, but you might ask yourself, well, why might you use them? Consumer acceptance and appeal is one of the number one reasons to think about using a biopesticide for the most part. They sound good. They are natural in many instances, and their environmental impacts are often much lower than considering a multi-site conventional. If you're growing organic, many of them are OMRI listed products, meaning that you can use them in an organic operation, such as my organic apple orchard that we have in Geneva. That's another option as well. If you have a good production operation, and I'll show these in action in some of my covered production trials right there. You've got strawberries here, double row, we got weed cloth, I got plastic, I got stuff overhead, I've got drip line. I've practically made a fake greenhouse. And they work really well in these type of situations, and you'll see when we start looking at some data. If the operation is small and you're agile, we saw with that NEWA model that, if you're using maybe a conventional fungicide or a single site fungicide, you may not have to spray as frequently. But many of these biopesticides, might require frequent applications, because just like the bottle of ethanol, they might go away very quickly, or they might break down, or they might wash off a lot more quickly than sort of a modern synthetic product, whether it's something like Cap 10 or even something like Pristine. And in many cases, you want to use these when the disease pressure is light. But if you use covered production, you can really reduce the potential threat of disease pressure. So in that instance, there's a couple of different ways these biopesticides work. Here's some really cool diseases. We've got white pine blister rust, got our standard strawberry anthracnose, got some powdery mildew, some leaf spots, and they work 3 different ways typically. There's an organism that's in the biopesticide that makes an antibiotic metabolite. And you're like, is this like an antibiotic that we might get from the doctor? Yeah, effectively it is. But what has not happened to it is they haven't taken that antibiotic out, resynthesized it, and adjusted it. These are just a natural one. And all the antibiotics we've ever had in medicine all came from organisms anyway. In this case, we're just taking the soup of those metabolites and putting it on a plant. And there may be more than one thing working instead of just one. But essentially, it's the same thing that we did to find streptomycin. We are just using the organism instead of a purified synthetic product. And what else do we have? Competitive inhibition. These work really well. There's usually a part of the plant, in this case, flowers. And if you have a flower infecting disease, this type works really well. If not, I wouldn't recommend it. And defense inducers. Well, these kind of turn on the plant's immune system. It's not as good as our immune system, but it can do some things to help really reduce disease. And I'll show you some trials that have all of these things in them at the same time. And so, you might ask, yeah, we've seen all these different mode of actions. How did the conventional fungicides work? They usually just kill for the most part. And in that instance, that's sort of how they work. They're always usually ones that just sort of kill the organism. They don't do competitive inhibition. They don't often turn on defenses. But just in case you're wondering how they all sort of play together, that's what it is. And if you want, for fun, you don't have to. We can go ahead and check out the little poll here, and you can try the first one. I think maybe I've activated it. You might ask yourself, do I use conventional fungicides? Do I just use biopesticides or organic, or some of both? Questions I always want to know of the different growers, and we will take a look, and I want to make sure. Oh, it is working. Yep. I use some of both. I use biopesticides. I use conventional. There's nothing wrong with any of them. I use some of both, and as well, depending on how things go, and you'll see how they work. Very cool. All right, I'll let you answer that booger, and then we will go through there. Always interesting to know these types of things. Yep. Very cool. All right, so let's talk about it. And some of the things I've done. You might ask, how well did these biopesticides and fungicides work on some of the diseases like gray mold and anthracnose and the gross leaf spots that sometimes appear to devastate your planting towards harvest? And talk about some of the diseases. We do a little bit of work on common leaf spot. It's very popular out there. It's got all these sort of circular purple, rust red type of lesions on the leaves with gray centers. And it can also cause something called black seed disease, where you can see this disease, seeds of this strawberry are really dark. And, it looks like it's got some other problems as well. But that was a nice photo that Meg McGrath had of black seed disease. And there's leaf scorch. Now we just purple blotches. A lot of these things all behave very similarly, and they can be managed a lot with the same stuff. The other ones that we're worried about as well are anthracnose, causing some really gross sunken necrotic lesions on the fruit shoots. We talked about this briefly in the other one. It makes a sort of spores if you get down in there. It might just look off colored there, but what's really in there is a lot of spores in many instances. And just like we mentioned earlier, it really loves rain. And even if you have cover, you get high relative humidity building up if there's a lot of rain outside the cover, and it can lock a lot of that in and still end up causing trouble. Gray mold, another very similar thing. Fruit rot, blossom blights. This is where it really starts around bloom. And you get all the nasty tufts of mycelium and spores on the fruit. And once again, it likes the same thing. Surprise, surprise. All of them love rain and they love moisture. That's why the bathtub gets moldy. Fungi are there and like, oh, great, bathtub mold. Water, yep, you don't have a fan blowing in your shower. So yeah, you have to clean it every now and then. And the same thing can happen too. And undercover, if it gets really wet outside or your irrigation's running all the time, it will accumulate a lot of relative humidity, give you things like powdery mildew, but in many instances allow these things to take a foothold. And you can see how this infection took place. The strawberry was touching the soil line, which is a very common thing to have happen. And so now let's talk a little bit about it. And I'm super curious. And as we go through the next thing, think about answering what your favorite fungicide is. I think we have this poll live, and I'm going to liven it up. What is your favorite fungicide? I'm going to activate that one. And this is a free form answer. You just can paste whatever's in there. And if you've always wondered what you and your friends are using, you can see if anyone knows a fungicide or wants to enter one. I think you can just enter anything that you want. Captan, it could be anything at all, and it will just make a little word cloud, and then you can enter different things. Could be sulfur, could be copper, could be anything you want, could be pristine. And then at the end I'll minimize it, and you can see if you have a favorite fungicide. Or I always like to know what people really like to use. I have my favorite fungicides as well, and we'll see them in this gray mold and anthracnose strawberry trials. And we did a bunch from 2018 to 2020. In many instances, we have a double row low tunnel planting from Dubois and day neutral. I do everything on Albion. Why do you only do day neutral trials, Carrick? Well, that's because we're spraying apples until July, and I can keep cutting the flowers off the strawberries until August, and so I can switch to spraying a different crop all the time. And so that's why the day neutrals are really popular. It allows us to really focus our attention all on strawberries when we get to this part of the year. And so this is one of the things that we did with the New York berry growers. They gave us an incredible amount of research funding, which has been awesome. And I was like, you know what I'm going to do? I'm going to test all the major AIs in the entire guidelines, and then we're going to see what happens. So I go out there and we spray these day neutrals right at harvest, maturity, and replicated plots with all these different things. Scala, Rovrol, Elevate. You can see one of everything. Captan, or the green ones I put are generally OMRI listed. White mineral oil, double nickel, Bacillus amyliquafaciens, Bacillus subtilis, Quava. I put a copper in there. I made it blue. And almost one of everything that we have. We have Pristine and Maravon in there. So I spray them all at harvest. I grab all the fruit, and then then we come back and then I inoculate them with the pathogens, which is a really nasty, hard test. It will rot fruit very, very quickly. And it won't tell you what you would get in a commercial level. They'll probably all work just fine, but it really sort of gives you a relative performance of all of them. And we did it for a couple of different years, and we found some interesting things. So this is what we have in the two graphs, and a lot of information can be found here. If you want to see this in more detail and reflect on it, you can go to the New York Fruit Quarterly. My student David published, I think, all of our New York berry growers research in the Fruit Quarterly for the last couple of years. And I think the fall episode, I didn't see it the last time I looked, but I was ensured that it was there and coming. Maybe it's only in the free episode, the free issue will show up on the website shortly after it's not the first one. But what you have is the incidence of strawberries right here with gray mold or anthracnose, gray mold and gray, anthracnose in that orange gross color. And over on the right is just how many fruit were clean. So you really want a high bar here. You really want low bars when you talk about fruit with anthracnose. And remember, this is a really hard test. Things rot really well. And for the most part, stuff will look pretty good if it stays below this 35% line. sort of my arbitrary value of this type of things. And what really works well, Scala works fine. It's a fungicide that was really designed to work on the botrytis, the gray mold pathogen. Roverol is an incredible powerhouse of a fungicide, and Elevate may not be as effective, but Switch has 2 different fungicides in it that is incredibly well. You can compare everything to Captan, and if it does just as good as Captan, you've got a pretty decent product. You're like, wow, look at that, JMS silent oil, organic, that can do as well. There's other things to be concerned about with that, but yeah, these products work. Wait, why is Pristine working so poorly in this particular instance? What we believe we might have as our isolates may have actually resistance to both the QOI and maybe a little bit of the SDHI product. The Pristine has one fungicide in it. It's an older generation of the SDHIs, while Maravon, which is also labeled for strawberries, and we're using the strawberry rate right there, has a different one. And you can see it just really makes the world of difference in that particular instance. And, it could be the fact that it's easier to mix. It's a liquid. But it also has that fluxaperoxide that I mentioned a little bit earlier as one of the AIs. And it just, sometimes the populations in New York just don't have the mutation to beat that particular one. But this one has boscalid in it, and it's historically a little bit problematic in both the gray mold and anthracnose pathogen. But it works pretty well, even maybe in against these resistant isolates. You can see, yeah, you can do fine with double nickel right there. Serenade Opti, not so much. We'll creep on down the line. Copper, a lot of the protectants and stuff may have a little bit more difficult time. Ronilin, I don't even know if Ronilin is labeled. I don't know. That's a good question. I've never used that one. Let's see. Inspire Super work really well in gray mold, not so well on anthracnose. And so I mentioned before, they're all the same type of fungi, but there are differences in how they can perform. And it's interesting to see how much gray mold was there versus Inspire Super. Fontellus was a real strong performer. And you can see the ones that really worked well. Inspire Super had a lot of clean fruit. And, these 2 bars add up. Any rot on a fruit is not good. And that's why I always like to show the fruit graph. And you can see that a lot of them do just fine. And maybe with a little hit on the Serenade Opti, it's a powder. I find the double nickel LC is a liquid, and I find that's a little easier to mix. But, Fontellus looked great over here, but for some reason there were other rots that were going after it. And I don't list all the rots, but they're there. Let's take a peek at 2019 data. Pretty much the same trends for the most part. We got a nice bump here with Maravon really working well. Not a good look for Pristine in the same year. I think it's resistance that's taking the issue out. You can kind of see Serenade not working so well and being one of the lower ones there, even though it did fine on anthracnose. And remember, fruits can rot from just about anything. Fontellus being a strong performer, Iprodione and Roverol being strong, and Switch being very strong in this case. And we know from a lot of research that we've done and published that there's very little resistance out there to the materials in Switch. You can see there are 2 things that aren't really in any of the other groups, and it's a unique opportunity for growers to have them. And sort of summarize what we found thus far is that Roverol and Switch are extremely effective. They're just rarely used, and they have rare use restrictions. Like Roverol can only be used maybe twice and only before bloom, or something of like that. So it doesn't give the opportunity for pathogens in our area and regions to ever get a lot of resistance to it. When you can use them, I would. If we rely on Switch all the time, yeah, we'll burn it out. So it's always good to have things like Captan and other protectants in there. I think with Pristine, I've got some resistance in the isolates we use, but that may not be the case for you. But QOI resistance and SDHI resistance is very common all the way, in Maryland and all the other big strawberry production areas. May not be affecting you, but I find that their Maravon product is just dandy, as well as the Fontellus. These have different members, more modern SDHIs in them that seem to be able to require different mutations than the ones that would be required that we may have had for Pristine for quite some time. And if you're doing organic, these things, double nickel, Serenade, and Quava, are all very viable options. And as we creep to the next area, I will show you. Now I'm going to ask a different question, and you can, oh goodness, let's see if we unactivate this one. And I want to curiously see if any of these, what's your favorite fungicide? Let's try this one. I'm going to activate this one. You can ask yourself now, Christina, do you think you'll change any of your fungicides that you use? I'm sticking to fungicides on the list, or maybe I'm already using some of these fungicides. So maybe they're already part of your plan. You're like, nope, I want to use my own favorite stuff. So you can answer those. It's A, B, and C. And I'm going to, while you're clicking and picking, I will just sort of talk a little bit about the setup for our covered systems trials. You might notice like I've got the sides pulled up there, and that's so that I can spray them. We just go through, we pull the sides up, and in this case, I use a wand, and pretend like I'm small farmer, and you just sort of go right down there, and I get fine coverage just sort of leaning my wand under there and walking the plots pretty well. So we have double row, day neutral, Albion strawberries. They're all planted under. In this case, we do cover versus no cover, and this is where you're going to see some weird things bouncing around. The fungicides are timed using the calendar or the NEWA model. We start at bloom, and then I run the program from like August through October. And, normally you could run day neutrals a lot longer under cover. At some point, I just have to cover them to just try to force them to quit making fruit, because I've got to start teaching again. So we just have to stop. And I look at the development of leaf spots, and I look at leaf spots and fruit rots. I mentioned before that, the models are really for fruit rots, but, you can see yourself how they ended up working. We got 5 minutes. The cover's great for a couple of different reasons. I don't want to spend too much time on it, but water and rain spreads disease. Cover's awesome. Wind spreads spores and disease. Cover's great. However, wind also reduces relative humidity and drying time, and this is where cover has a trouble. So you want to have a little bit of movement through there. In often cases, I leave one of the tunnels open like a quarter on one side. It keeps the rain off, and then it gets a little bit of air circulation in there on those low tunnels. Soil contains moisture, and soil moisture spreads disease and initiates root infections. Yep. Yeah, I do that. I do exactly what Laura put in the chat. We find that very thing. It's like, oh, we're gonna have a big rain. Go put the one side down, but I do like leaving one up. And then weeds harbor pathogens, so you still have to worry about weeds in cover, but they're a great stage for biopesticides. So one of the trials that we did, these are kind of a little bit complicated to look at, and the pressure varies from year to year depending on how much rain and water we get. Even if it rains outside, there's a little more pressure in the tunnel. Rains a moderate amount, things are a little bit different. You can see all of this in the New York Fruit Quarterly articles as well. So we have program by timing, and this is gray mold at harvest. And I use a program of Elevate and Switch, two pretty good fungicides against double nickel alternated with Lifeguard. And Lifeguard is one of those ones that boosts plant defenses. No biocidal activity directly. And this is the NEWA bar. Set of graphs here. You can think, so this is organic versus conventional. Yeah, conventional did a little better. And this is the calendar bar. And you're like, well, look, the NEWA set of bars is very similar to the calendar set of bars. That's kind of cool. You might notice that there is a good improvement, but this year was a light year. And you're like, well, wait a minute. Okay, I can get by in a light year with 6% disease versus 10% disease. Sure. Okay. In this case, yeah, there's a statistical difference. But I could live with that. I mean, 6 versus 10% disease at harvest. I mean, I get 10 rotten fruit just breathing on them. But look what we've noticed here. These are the threshold dates. If I just went every week, bam, bam, bam. You often spray on sometimes where it's not really that useful. And there's no difference in the two sets of bars. And I only sprayed 3 times that entire season when I sprayed on the NEWA. I still had to go out for the calendar, but that's pretty decent. 2019, this time we had a heavier, a harder year, and fruit rots are really hard for the biopesticides. Now you can say, yeah, okay, in this big wet year, I really got, the conventionals weren't too bad, but you don't have to do one or the other. You could do a little bit of each. You can see now we had to do more NEWA applications to really get it, but it's 5 versus like 10 or something of that nature. So that's not too bad at all. 2020, looking at some gray mold and stuff like that. This year was a weird year. We had some dry year, but fruit rots got a little bit problematic. There's a lot more variability. One thing you'll notice about the biopesticides, these error bars tell you how much error is associated with my replicated plots. If they're really tall, there is a lot more variability, but look how tiny they are with a conventional. There's just more consistency. I mean, it's not a big difference in fruit rots in general, but This was anthracnose or gray mold in this case. But at this time, the NEWA timing is much better. And, between NEWA and between calendars, not that much difference. And very briefly, we also look at leaf spot trials. I'll just sort of go through it in this particular instance where I'll just sort of show you one and then we'll end it. In this case, we found cover was actually higher. This is cover, no cover, and this is how much leaf spot than an open area. It may have been a really high relative humidity that year. So that was very interesting. But the conventional wasn't really that much difference and for leaf spot. And in this instance, you're not really with NEWA, you're not getting that much more leaf spot than without. Actually, with NEWA, we got better leaf spot control, even though the model is not made for it. And that's some of the types of things that we see. And we didn't have any in 2019, oddly enough. That's a really strange thing to imagine. But here, same sort of thing, cover, no cover. In this case, we got a lot more. This was a really dry year, and I think the cover prevented anything from landing on. And so we didn't have a lot of, our drip was probably better managed. In this case, less leaf spot and cover. Very crazy stuff. But for the most part, NEWA was a little better for keeping my strawberry leaf spot down than using a calendar application. And I think that's one of the more interesting things that we talked about. I think you can use biopesticides and organic if you use them with forecasting models and you use them with cover production. And, they're really good. And, maybe the best for practice is when it's dry and I don't need it that often. That's when I use my biopesticide. That's one of the research projects we're doing in apples. And I think we're going to do that with our berry grower money this year, is to try to use the more integrated one. If it's really going to be wet and rainy, we might try switch. If it's not, then I might go double nickel. And everything has really been funded directly by the New York Berry Growers Association. I'll take any questions and then you can do your polls, etc. I'm going to go ahead and stop this share. Very fun.
All right. Thanks a lot, Carrick. That's a ton of
Welcome.
information and
Yep.
some great information about efficacy, which is something that people are constantly asking me about. So I appreciate it.
Yep.
I do have one question about Fontellus. There's
Yep.
some people that are not sure that, and I'm not sure if it's yet legal in New York State.
I'll have to look on that one. Now, that's a good question. Let's take while we're asking some other questions and you're running a poll, I'll look it up. I thought I looked before I came, but I might not have looked that one. Might have got focused on Maravon. So what we're going to do is, while you're going through the next question, I will sort of give a peeky poo and take a look.
Yeah, it's been one of the, that is a chemical that's really used a lot for fruit growers. So it would be great if it was.
Yeah, for some reason I thought I saw it on strawberry, but now you see that this is really slowly working right here. Well, we will take a look. What are any other questions?
Actually, I don't, unless they might be texting you or chatting you privately, but let me just see.
I think I saw it. It's got to check all the, well, let's try the primary one too. I think I saw it the other day. Let's see. I'm going to do this. One of the cool things you do when you download it, now that they've got it fancier, is you can type the word strawberry. Oh, there's seven mentions of the word strawberry. Well, where are they? Yeah, strawberry and low bush. There it is. You can use it for botrytis, fruit rot, gray mold, and so and so. Now, the one thing that I have noticed about all these really fun and fancy products is they're not allowed to use on Long Island. Let's see, the restricted use, and I'm going to go back to this page. Oh, I don't see the anti-Long Island. Nope, no use on Long Island. Yep, that's where it gets really sad. And so I have to go give a talk to Sandy's group on Long Island. I'm going to just constantly be saying, Maravon, yeah, I showed you all this data that works a lot better, but you can't have it. And then you'll just boom, boom,
well, that's great to know about Fontellus. That's great news.
Because right now you don't see it here in this label, but it's on the other thing. And that's where you always, another thing you do is you just type in long, and you're like, oh no, long sleeve shirt, long pants, long bean. I don't see the restriction on the actual label, but I do see it on their webpage.
For Long Island, you mean?
Yeah, I mean, yeah, Long Island. I mean, just like I'm looking up the word. Yeah, I mean, maybe that part is land. Yeah. I don't see it on the actual label, but it's staring me right in the face right there. No use on Long Island. Huh. That might be something worth asking DEC about, because that would be cool for those particular growers. Same with Maravon. All the new stuff, it's all no Long Island. Super sad.
Yeah. Well, that's great to hear for the rest of us upstaters. So
Yep.
that's good. And I did put the link to Sandy Menasha's Suffolk County Extension is sponsoring a virtual workshop that will be on March 25th. And if you're interested in more strawberry information, Carrick will be doing a related, slightly different talk. And there's
Fruit
several other
diseases, I think.
speakers.
Yep.
Yeah. So that might be another way. And I know they have DEC credits if you're looking for more credits. And that brings me to the other question that somebody had about when you'll get the credits. We'll be sending out actually you have the credits pretty much now as soon as we send the roster in, but we will also send you a certificate for your own files. DEC is no longer requiring them, but it's really good to have just in case, as I've already heard several times, people, the DEC rosters sometimes are not exactly right. And so it does kind of helps them go back and check the rosters. Yes, and Larry, yes, people can, we're officially over. So if you guys need to log out, we've got your names, we've got the poll information. I'm thanking Carrick very much for being here and giving two great presentations. And Natasha and Chelsea and Andy, thank you guys for your help in supporting these virtual workshops. And yeah, everybody else, if you're going to come tomorrow, it's the same kind of thing. We've got tomorrow at 7:45. If you want DEC credits, try to log on a little early, and then we'll get started right at 8 o'clock, be done by 8:30 tomorrow and Thursday. So